As some of the speakers have alluded to, and as I think everybody knows, dopamine is the key neurotransmitter that's deficient in Parkinson's disease. But in addition, it underlies many of the debilitating symptoms of ADHD, schizophrenia and drug addiction. And Dr. Tim Oman, who is from Melbourne but could not be here tonight, has found in an animal model that the number of dopamine cells, the number of cells, not their function, can be responsive to environmental influences, and they wish to pursue that topic further, and will do so in non-human primates, in monkeys, looking to see whether environmental enrichment can change the number of anatomical cells in the brainstem of those monkeys, and then go to humans who and obtain brains, actually from Edinburgh, where they know that the patients have died during, they're asking the question, does seasonal variation alter the number of dopamine cells in the brainstem? And they'll obtain from Edinburgh a brain and tissue bank from Scotland, 10 brains of patients who have died, they lived at high latitude and died during summer versus winter, to see whether this is also a phenomenon that can be extrapolated to the human condition. However, the next project in the Parkinsonian area is from Dr. Rebecca Nisbet from the University of Queensland, and I hope she will explain to us what tau and tau antibodies are.
A bit of a walk in high heels, sorry. First and foremost, I would like to thank the staff and directors of the Brain Foundation, the Scientific Advisory Committee, the fundraisers and those who have generously donated to the Foundation. As an early career researcher, it's quite difficult to get your foot in the door and start to receive funding for your own research. So I'm extremely grateful to be standing here accepting this award tonight. There are currently over 200,000 Australians living with Alzheimer's disease, and this number is expected to double by 2030 if there continues to be no effective treatment. One of the main pathological causes of the disease is the aggregation of the protein tau within neurons. We all produce tau over our entire life, and its normal function is to stabilise the neuron's transport system. In Alzheimer's disease, however, tau is modified by a process known as phosphorylation. This causes tau to aggregate into tangles and results in neuronal degeneration. This has prompted our group at the Clem Jones Centre for Ageing Dementia Research, under the direction of Professor Jürgen Gertz, to develop therapeutics aimed at inhibiting tau phosphorylation and prevent its aggregation for the treatment of Alzheimer's disease. The challenge, however, is to generate molecules that can get into the neuron where tau is localised. To overcome this, I'm currently developing small intracellular antibody fragments called intrabodies, which can be delivered as a gene into the neuron. These intrabodies are not only designed to specifically bind to phosphorylated tau, leaving normal tau to function, but they're also engineered to clear pathogenic tau from the cell by taking advantage of the cell's protein degradation machinery. The funding provided by the Brain Foundation will allow us to test the ability of these intrabodies in clearing pathogenic tau and reducing cognitive decline in mouse models of Alzheimer's disease we have established in the centre. It is hoped this work will provide a strong foundation for progression of these intrabodies to human clinical trials. Thank you very much.